Hi all, today I'm going to be presenting my thesis work examining region and context-specific ensemble dynamics generated following administration of methylene dioxymethamphetamine. 3,4-methylene dioxymethamphetamine, colloquially known as MDMA, has gained considerable traction as a novel therapy for treatment-resistant psychiatric illnesses such as post-traumatic stress disorder and autism spectrum disorder, given its ability to enhance sociability. Yet, MDMA displays both social and non-social reward, perhaps explaining its potential abuse liability. While MDMA's prosocial effects require serotonin release in the nucleus accumbens, its non-social reinforcing properties are mediated by dopamine release in the same structure. Yet it is unknown how these behavior-specific neurochemical actions relate to circuit physiology across the brain. So, using transgenic mice enabling activity-dependent neuronal labeling, we examined the region and context-specific characteristics of neuronal ensembles generated following systemic MDMA, diphenfluramine, or methamphetamine delivery in social versus non-social behavioral contexts. To begin, this study involved the use of the TRAP-AI14 biogenic line, which fluorescently labels recently active neuronal populations in rodents during a confined experience. This system relies on one transgene expressing CREERT2 under control of the FOS promoter, an immediate early gene displaying elevated expression following neuronal spiking, and an additional transgene expressing an effector gene in a CRE-dependent manner. In this case, TD tomato under control of the constitutive promoter ROSA26. Notably, the presence of hydroxytamoxifen, or 4-OHT, and neuronal activity are preconditions for trapping. In this way, neuronal activity enables activity-dependent expression of CREERT2, while 4-OHT induces recombinase activity and expression of the fluorescent reporter TD tomato to visualize ensemble dynamics. For each of our experiments, trap mice were either housed in isolation or with an age and sex-matched conspecific. We then performed 3 days worth of habituating saline injections, followed by synchronous 4-OHT and MDMA, methamphetamine, or diphenfluramine injections. Following 1 week of expression, we then perfused and retrieved acute coronal slices for imaging and TD tomato positive cell counts. Below, we have our regions of interest, which correspond to MDMA-induced hotspots our team has previously identified via whole brain immunolabeling for FOS. The far left represents the NAC medial shell, the middle the NAC lateral shell, and the far right the extended dorsal interpiriform and claustrum subregions. So here are our results following MDMA delivery in social or non-social contexts. And I want to focus specifically on the results in the NAC medial shell and DENCL. As shown on the bottom left, MDMA induces TD tomato expression in the NAC medial shell that is more robust in social versus non-social conditions. This elevated expression results in greater labeling across the medial shell while stretching marginally into the accumbens core. The same pattern in TD tomato expression is true of the DENCL, as shown on the far right. Mice injected with MDMA in social conditions seem to display a greater density of TD tomato positive cells at the intersection of the ventral and dorsal claustral subregions. Lastly, while mice injected with MDMA show more robust TD tomato expression in the lateral shell compared to saline controls, the abundance of labeled cells is comparable across social environments. So given MDMA's broad pharmacological actions, we sought to examine whether compounds that preferentially interact with specific monoamine transporters can also replicate observed patterns in TD tomato expression. Diphenfluramine and methamphetamine offer a means to do so. The fenfluramine is a substituted amphetamine with mechanistic similarities to MDMA. This compound, however, acts as a selective serotonin-releasing agent. Like MDMA, fenfluramine delivery also enhances social preference among rodents while displaying limited acute reinforcing properties. By contrast, methamphetamine primarily interacts with the dopamine transporter to augment dopamine release. Unlike MDMA and fenfluramine, however, methamphetamine administration does not appear to increase rodent sociability. The drug is also acutely reinforcing. So given these chemicals' unique properties, we can leverage them to refine our understanding of social modulated ensemble dynamics in the NAC medial shell and DENCL. So as before, we performed the same experimental paradigm, but instead delivered methamphetamine or fenfluramine in conjunction with hydroxytamoxifen. Here we have our results among mice injected with methamphetamine in social or non-social conditions. In the medial shell, meth-injected mice show elevated TD tomato expression compared to saline controls regardless of their housing condition. The same is true of both the lateral shell and DENCL. Yet only mice injected with the drug in social conditions show a significantly greater abundance of TD tomato positive cells. In this way, it does not seem that meth induces ensemble dynamics sensitive to the social environment, and this finding is consistent with its behavioral effects. Next are results among mice injected with fenfluramine in social or non-social conditions. So interestingly, Fenfluramine delivery replicates patterns of TD tomato expression observed in MDMA-injected mice. In the medial shell, fenfluramine administration generates the most robust TD tomato abundance among socially housed animals. The same is true of the DENCL, but not the lateral shell. MDMA-mediated FOS expression in the NAC medial shell and DENCL thus display sensitivity to the social environment in which the drug is delivered. This effect is mimicked by fenfluramine, but not methamphetamine. And considering the unique pharmacological mechanisms of each compound, our study suggests a unique role for serotonergic signaling and social modulated ensembles in both midbrain and cortical structures. And in describing the anatomical and context-dependent features of distinct MDMA ensembles, we set the foundation for broader functional studies. Thank you.